Good morning, everybody, here in person and online. I'm Alexandra Sasha-Slavkovic, Professor of Statistics and the Associate Dean for Research in the Eberly College of Science. It is my honor to welcome you all to the fifth week of the 2026 Ashtekar Frontiers of Science lecture series. This popular public winter series is now in its 32nd year. It was founded by Abhay Ashtekar in 1995, soon after he arrived at Penn State as a director of a new research center that is now the Institute for Gravitation and the Cosmos. It owes its successes to tireless efforts and meticulous planning by Barbara Kennedy, who presided over the series during its first 25 years, making it one of the most successful science outreach events in central Pennsylvania. This year's lecture series, titled Unexpected Pairings, Addressing Today's Biggest Societal Questions Using Different Disciplinary Approaches, will showcase how researchers from across the college's 7 departments leverage wide-ranging methodologies and theoretical approaches to tackle some of today's biggest societal questions and pressing scientific issues to help shape the future of science, health, and humanity. Each of the 6 lectures features 2 high-level overview talks on topics including origins, medicine, resources, materials, climate, and education. If you missed one of the previous weeks, you can find the recordings at science.psu.edu/frontiers. Today's topic is resources. The first lecture, titled, What do plant cells' walls look like and what can we make from them, is being presented by Charlie Anderson. Charlie is a professor of biology and associate head for research and faculty success in the Department of Biology at Penn State. His group uses advanced microscopy, cell biology, molecular genetics, and biochemistry to investigate the dynamics of plant cells' walls with the goal of informing efforts to sustainably produce energy, food, and biomaterials from plants. He also chairs the Plant Biology Intercollege Graduate Program, serves as a theme lead for climate and natural systems in the Institute for Energy and the Environment, and co-directs Penn State Center for Biorenewables. The second lecture, titled Mining and Separating Rare Earth Elements Using Proteins, will be presented by Joy Cotruvo. Joy is a professor of chemistry. His lab works to understand how biological systems achieve selective recognition of metal ions and to develop biotechnologies for sensing, recovery, and separation of valuable metals, especially rare earth elements. His fundamental discoveries and translational impact of Joy's work have been honored with several national and international awards, and intellectual property generated in his lab has been licensed by multiple startup companies. He's actually started multiple startup companies, right? Joy. Following the lectures, graduate students Elisa Chernikova and Wonsok Choi will lead a moderated question and answer session with Charlie and Joy. So without further ado, Charlie.
Thank you so much, Sasha. It's a pleasure to be with you all this morning to talk about some of the science that's going on in my lab and to think a little bit about resources. So I'll start off by just highlighting that we're going to talk about 2 classes of biomolecules today, carbs and proteins. So if you had a good breakfast, you ate probably some carbs and proteins, and you're already on your way. All right, so my group here at Penn State is focused on understanding what plant cell walls look like, how they're built and modified during plant growth and development, and what we might be able to make out of plant cell walls, which are a very abundant resource. This is a really nice excerpt from a YouTube video by Drew Berry, where the authors were attempting to model what was happening at the molecular scale in plant cell walls. So the desks that you're all sitting behind are made of plant cell walls, or at least simulations of plant cell walls. And what I'm going to ask you to do is to think about going into the nanoscale. So we're talking about things happening at the levels of nanometers, right, which are millions of a human hair's width. All right, so you're going to join me in the nano realm. I will start off with a bit of a sobering statistic, which is that our burning of fossil fuels, right, humanity's burning of fossil fuels, is polluting our air. And that ends up killing more than 9 million people per year around the globe who would otherwise be able to live healthy and productive lives. This is a scientific fact that's been backed up by a lot of research. And if we look more locally here in Pennsylvania, you can see that our air quality is not as great as we might think it is. Even in Centre County, recently, the state of our air was graded a C by the American Lung Association. So I personally like to breathe. Breathing is great. I had the flu a couple of weeks ago, so I'm really appreciating the ability to breathe, and I'm sure all of you do too. However, I would posit that this is a solvable problem. Air pollution due to fossil fuel combustion is something that we know how to deal with as a society, and that's one of the things that we're focused on in the science in my lab. So as a cell biologist, I'm inspired by the ability of plants to take sunlight, water, and some nutrients from the soil and convert that into themselves, right, to build their own bodies and to generate a lot of things that are quite useful to us. So this is some satellite imagery zooming way out rather than way into the nanoscale this time. We're looking at the face of the planet. And you can see that large parts of our planet are blue, right, the oceans. They also actually contain many photosynthetic organisms. But a lot of our land area, including here in Pennsylvania, is covered with plants. I like to teach via the Socratic method, so I'm going to ask somebody from the audience, does anybody have a guess for how much plant biomass exists on Earth? Any guesses? I'm staring at my own children who are in the audience to see if they have a guess. Anybody want to give a guess, a number? I'll give you a clue. It's in billions of tons. People are feeling a little shy this morning. Maybe you were all up dancing at Thon last night, so that's why you're a little tired. It ends up being about 900 billion tons. So almost 1 trillion tons of biomass is embodied in plants. It's a huge amount of resource. And if we think about how that plant material can be used by society, right? Humans for thousands or even millions of years have used plants to produce food, energy, clothing, and materials. And I'm going to touch on some of those uses and how we're advancing the science of how we might be able to use those in more sustainable ways later on in my talk. One of the things that's also inspiring to me as a plant biologist is the idea that using plant cell walls, right? These are the components of plants that make up most of their bodies to generate energy and materials might actually result in both lower pollution, because we're generating types of fuels that don't produce all of the air particulates and sulfur dioxides and nitrous oxides that are so damaging to our climate and our air quality, lower overall air pollution, and also net negative greenhouse gas emissions. So if we look at this graph that was published several years ago by the US EPA, gasoline, this is just standardized to 100% of the greenhouse gas emissions that would be produced by things like gasoline and diesel. Right now, most of the biofuels in the US are produced from corn. It's corn-based bioethanol. So if you go to the pump and you see the sign saying, about 15% of this fuel is ethanol, most of that is coming from corn. And we do get a benefit from that corn ethanol in a reduction of about 20% greenhouse gas emissions, accounting for all of the production, the transport, and the burning of that fuel relative to petroleum. So that's good. In places like Brazil, they use sugarcane to make ethanol, and that's even more effective. They get a 60% reduction in greenhouse gas emissions. But if we were to use something like switchgrass, which is a native prairie grass in North America, to generate ethanol, transport that ethanol, put that in our tanks, and burn it, we would actually end up with net negative greenhouse gas emissions. And you might be asking yourself, how is that possible? Because the carbon is captured by the plants via photosynthesis. We then convert that using some energy and processes into ethanol, and then we burn the ethanol. Wouldn't the lowest you can go be 0? The reason it's negative is because plants don't just produce above ground parts. They produce roots. So you might have heard about some technologies that are called direct air capture for carbon sequestration. Plants are doing this all the time, essentially for free. They capture CO2 from the atmosphere, they convert it into sugars that they transport throughout their bodies, and some of those sugars are transported down into the root systems. So they are burying carbon underground, and a lot of that carbon is very stable and stays in the soil for hundreds of years. And again, they're doing this on gigaton scales, multiple hundreds of gigatons per year around the globe. So plants are carbon capturing machines. So in my group, we try to understand the nanoscale architecture of the plant cell wall. And we try to understand how that architecture changes over time. We think of the plant cell wall as a dynamic system that allows plants to grow from a tiny seedling to something like a large tree. And in understanding this assembly, we build conceptual models of what the plant cell wall might look like. This is a classic model from about 20 years ago, in which we think about how cellulose, which is the physically strongest and most abundant component of plant cell walls, is arranged in that cell wall at the nanoscale. So you can see that the cellulose, which is in green here, is laid down kind of like cables or even like rebar and concrete. It's really reinforcing the structure of the plant cell wall. Cellulose actually does have a tensile strength that's similar to that of steel, and it's arranged in a linear fashion to give strength to the cell wall in ways that we're still figuring out. That cellulose is surrounded by other polysaccharides called hemicelluloses and additional sets of polysaccharides called pectins. Pectins are some of my favorite. If you've ever had jam or jelly, you've eaten a bunch of pectin. And these collectively are called matrix polysaccharides. In addition, there are proteins called glycoproteins that are heavily glycosylated. They have a lot of sugars attached to the protein core. And all of this together makes up this relatively complex structure of the cell wall. This model is actually 30-fold less dense than we think the cell wall really is, right? So it's a very dense network of polysaccharides that's making up this biomaterial. One of the things that we do in my lab is we like to watch plants actually assemble their cell walls in real time. And I'll introduce one of the really important protein-based machines that makes a component of the cell wall called cellulose. This protein-based machine is called the cellulose synthase complex, and it moves along the surface of plant cells through what's called the plasma membrane and spins cellulose into the wall in a way that's somewhat analogous to a spider spinning silk. And as it does so, it's guided by another protein-based structure called a microtubule that sits just under the plasma membrane and attaches this cellulose synthase to the microtubule and allows it to produce cellulose in a linear fashion, which is how cellulose is partially organized in the wall. So this polymerization of cellulose out of the cellulose synthase complex actually pushes that complex forward as it's moving through the plasma membrane. So we think that's what provides the force that causes this complex to move. One of the great things we can do here at Penn State is take advantage of advanced types of microscopy. So we used a super resolution microscope that we purchased here at Penn State back in 2016 to image cellulose synthases in living plant cells. And this is just a portion of a plant cell, as well as those microtubules. So you can see in green in this time-lapse movie, small particles that are transiting along the microtubules, which are in magenta. And this is data that was collected by Sydney Duncombe, a former PhD student in my lab, in which we are trying to understand, again, the nanoscale details of how plants are putting together their cell walls and organizing the cellulose. So by doing studies like this, we can understand cell wall assembly a little bit better and advance our models and understanding in a way that allows us to potentially take advantage of cell walls as a resource. So what can we use cellulose for? Cellulose can be used to generate filters that remove pathogens from our drinking water. This is pretty important since none of us like to drink water that's filled with pathogens. We can generate recyclable and compostable food packaging. So I'm sure all of you have had the experience of getting maybe a takeout, dinner, and you end up with a bunch of plastic. And you're like, what do I do with this plastic? If we can generate that same type of packaging that's durable and yet recyclable and compostable using things like cellulose, I think that would be a benefit. And then importantly, on the biomedical side, cellulose can form a scaffold for tissue regeneration and regrowth. So we can harvest cellulose from resources like trees and switchgrass and convert that into a technologically advanced scaffold in which we can grow human cells to do things like skin tissue re-engineering and repairing our own bodies using that cellulose as a scaffold. Because animal cells like to grow in a scaffold, often of some kind of extracellular material, things like collagen, or in this case, cellulose. So I'm going to highlight one story today, which is relevant because we are actually in the building where this story was generated. The microscope that we used for this story is actually right around the corner if you walk outside. So researchers for a long time have understood that we can make fuels and other useful products out of plant cell walls. And one of the sort of jargony terms for plant cell walls is lignocellulose, because two of the major components in plant cell walls are cellulose and lignin, hence the name lignocellulose. So the concept is you take some kind of biomass, whether that's trees or switchgrass, you harvest it and transport it to what's called a biorefinery. It is then ground into small particles and treated with sets of enzymes to convert the sugars that are present in that biomass into soluble sugars, which are then fermented using microorganisms to generate fuel. And this is mimicking in a large way the process by which corn-based ethanol is made in the US today. Instead of needing to apply lots and lots of enzymes, instead the biorefineries for corn-based ethanol just use amylase, which is the enzyme that degrades starch into glucose. However, the challenge for lignocellulosic biofuels is that to do this, we need a lot of enzyme, because over 400 million years of evolution, plant cell walls have evolved to be very resistant to degradation, right? Fungi and bacteria and other things in the environment are constantly trying to eat plants, and plants have evolved resistance to that predation. And so the cost currently of the enzymes that are needed to produce lignocellulosic biofuels is about 50 cents per gallon of ethanol. And that makes the price of this ethanol high enough that it's not competitive with fossil fuels and corn-based ethanol. So that's one of the economic challenges in producing lignocellulosic biofuels. So there are sets of enzymes that are required to degrade cellulose, which is a highly crystalline substance, into its component sugars. I'm not going to go into the details of all of these enzymes, but I'm going to highlight one, which is an enzyme called CEL7A. It was discovered in a fungus called Trichoderma ricei, which in turn was discovered during World War II when soldiers fighting in the South Pacific noticed that their canvas tents were disintegrating, essentially. And it turned out that there was this fungus, CEL7A, that produces this cellulase enzyme. The enzyme has a catalytic domain that is able to extract a chain of glucose into its sort of tunnel and break that glucose chain into small components that are called cellobiose. That's a dimer of glucose. And then it also has a linker and a carbohydrate binding module that we think allows the enzyme to stick to the cellulose substrate. So we built a microscope. This is a collaboration with Will Hancock, who's in the biomedical engineering department here at Penn State. And we built this microscope that allows us to image individual cellulose degrading enzymes that are labeled with fluorescent quantum dots. So quantum dot technology is amazing. It's a silicon-based technology that generates these dots, these small 15 nanometer diameter dots, which are highly fluorescent and we can track over time. And we lay down some cellulose that's actually made, in this case, by a bacterium on the surface of glass. And we can then image. So that cellulose, in this case, is showing up as dark in this particular micrograph. And we can image these brightly labeled, quantum dot labeled cellulase enzymes as they land on the cellulose and start to degrade it. So you can see that over time, the density of these fluorescent dots is increasing on the cellulose. And if we then look at sort of an accumulation of all of the CEL7A enzyme in relation to the cellulose, as we can see it on the microscope, you can see that those two things are lining up nicely, meaning that our enzyme is specifically binding to cellulose. We can actually use tracking technology to track the motility of these enzymes as they sort of chug along and degrade the cellulose at a very slow rate. And by doing that, we discovered that a lot of these enzymes sit down on the cellulose and don't do a whole lot else. They don't seem to move. They don't seem to degrade cellulose. They spend a lot of time in what we call the stuck state. And by calculating the enzyme kinetics, transitioning between the enzyme in solution, it binding in a stuck state, it binding in what's called a processive state where it's actively degrading the cellulose, we can see that the propensity is more for the enzyme to be in the stuck state, which is not so wonderful, right? That means that about 90% of any enzyme that we add doesn't seem to be doing a whole lot to degrade the cellulose. So we're currently doing some experiments to see if we can engineer these enzymes to spend less time in this stuck state. And that in turn could increase the efficiency of that enzyme to degrade cellulose, therefore making the cost of lignocellulosic biofuels cheaper and more competitive with fossil fuels and other forms of biofuels. One of the downsides of that corn-based ethanol I've been mentioning is that it increases the price of corn. And we use a lot of corn to produce ethanol, we use a lot of corn to feed animals, and we can also potentially use corn to feed ourselves, right? I always love eating sweet corn in the summertime here in Pennsylvania. So the hope is that by using non-food-based biomass to produce biofuels, we might be able to avoid that food versus fuel dilemma. I'm just going to spend the last few minutes highlighting a few more stories of research that we're doing in my lab using plant cell walls as a resource. One of the things that we're doing is studying and engineering another form of cell wall called seed mucilage. So if you've ever had like a chia seed smoothie or a shake, you've probably seen that when you get the chia seeds wet, the sort of surrounding coating of the seed swells up and becomes kind of gelatinous. And that stuff is called mucilage. And Annalika Dutta, who's a PhD student in my lab, has gotten interested in this mucilage as a biomaterial. And she's done some really nice light sheet microscopy using a microscope that's right under the Millennium Science Complex building here at Penn State to analyze the structure of that mucilage. So in this micrograph, the cellulose is in blue, and we're actually navigating through the mucilage. It's like we're flying through the 3D view of the mucilage. And you can see that that cellulose is arranged in rays that extend out from the seed to the surface of the mucilage. And we think that that structure allows mucilage, in some cases, to cause the seed to attach to something like soil. And that attachment actually prevents the seed from being eaten by things like ants. So in the upper right, you can see a picture of a harvester ant attempting to pull a seed that's been stuck down with mucilage to a soil substrate. And the ant is really struggling to do that. It's like the old trick where you glue a quarter to the sidewalk and watch people try to pick it up. It's basically the same thing. And so we're trying to understand how this mucilage works from a structural level. And we're also carrying out what we call mucilage transplantation experiments, because not all plants produce seed mucilage. We think that by taking things like chia mucilage, transplanting that into the seeds of trees, we might be able to help reforestation success be a bit higher, because there are strategies right now where people are basically using drones to spray seeds over the landscape. A lot of those seeds don't survive, unfortunately, and we think that by adding mucilage, we might be able to help them survive to a higher degree. I'll also talk a little bit about wood, which is one of my favorite forms of plant cell wall. Wood is a very hierarchically organized and highly ordered material, right? So one of my hobbies is I like to play guitar, and I'm always amazed by how the different woods in the guitar give different sounds and different timbres. And part of that is how that wood is organized. So again, zooming into the nanoscale, we have things like cellulose and hemicellulose and this waterproofing material called lignin that wraps around that cellulose and hemicellulose and makes the wood very tough and resistant. And this is all organized into what are called macrofibrils. These macrofibrils are in turn arranged into different layers of the cell wall in something like a fiber cell, which gives many plants their upright stature. And then those fiber cells are arranged into tissues, which are in turn arranged into tissue layers and organs, and ultimately the body of a tree, right? So we're going all the way from 1 nanometer to 1 meter, or even in the case of large trees, 100 meters. So we're crossing a lot of spatial scales here. And by understanding this organization, we think we can engineer wood to do some amazing things. So we've been inspired by some researchers at the University of Maryland who invented a substance called superwood, which is basically natural wood that's chemically treated and densified. And that forms a substance that has many of the properties of a very strong metal. So it has, for instance, a higher specific strength than many aluminum alloys and even some steel alloys. And we think that's because of that hierarchical organization of cellulose and cells in the wood tissue that gives us this incredible strength when it's compressed into a dense material. And so Elisa Chernikova, who's sitting in the back and one of our moderators for the Q&A, is studying this superwood from a biological perspective. She's recapitulated this sort of densification, and you can see a block of wood on the left that's not densified. One on the right has been densified using what's called a carver press at 70 megapascals, if you like numbers, and about 100 degrees C, so about the boiling temperature of water for an hour. And we end up with this very dense substance that has high strength, but importantly also has a difference in its organization of cells. So Elisa has been doing some microcomputed tomography experiments at Argonne National Lab out in Chicago to understand how the organization of the cells in this wood change between the non-densified and the densified state. And we think that by understanding this, we can invent new forms of superwood and new strong biomaterials from this abundant resource. And then finally, I'll finish up with a story about generating food from plant cell walls. So I'm sure you all like to eat lots of fiber in your diet, but if I gave you a bowl of sawdust, you might not be so enticed by that. And it turns out that our guts are not adapted to be able to degrade lignocellulose. So one of the projects that we're working on in my lab, and this is a project that was spearheaded by Niroshan Siva, an assistant research professor who's sitting in the back row. So you can ask him all the tough questions about this project. We're trying to understand how we might be able to convert lignocellulosic biomass into edible food and do this in a way that will provide nutrition to people under conditions where conventional agriculture might not be possible. So one of the examples of this is if there were an asteroid strike, which there have been in the history of the Earth, right? That's one of the things that we think led to the extinction of the dinosaurs. If there were an asteroid strike, what would we eat? Because that asteroid strike would block the sunlight, right? It would put a lot of soot into the atmosphere, block the sunlight, prevent us from growing our normal crops. I mentioned earlier that there's 900 billion tons of biomass, right, on the planet, which is quite a bit. And that 900 billion tons of biomass is enough to feed everybody from a caloric standpoint for over 100 years. So there are plenty of calories out there. It's just that we're not good at getting them and digesting them. And so what Niroshan is doing is pursuing some strategies to take that biomass, convert it into things like acetate, which we can feed to algae. Algae, it turns out, are really good at metabolizing acetate and converting that into lipids and proteins, right? Because that biomass is carbohydrate rich. It has plenty of carbs. We can actually enzymatically degrade it into glucose. But if we're just eating glucose, I'm sure you've all experienced that at various times in your life, Halloween, things like that, right? When you're just eating a bunch of glucose, it's okay, but you wouldn't want to have that diet for the rest of your life. And so the thinking is that we can produce these other parts of the sort of nutritional balance for humans using things like algae and mushrooms to sort of balance out the calories that we would get from lignocellulosic biomass. And it turns out that strategies like this are already being implemented in the real world. One of my colleagues at the University of Cambridge, Paul Dupree, has helped start a company called the Supplant Company, where they take lignocellulosic biomass, partially degrade that into oligosaccharides, and then incorporate that into products like chocolate, which everybody likes. This chocolate tastes the same as regular chocolate. I've tasted it myself, but it has a lower glycemic index and is a little bit healthier than regular chocolate. So I encourage you to check it out if you find it in the store. And our hypothesis here is that biomass-based foods, right, lignocellulosic-based foods, might both enhance human health by providing a little bit more fiber in our diet and changing the nutritional balance in a positive way, and provide flexible sustenance for when, again, conventional agriculture, which has been so abundant for humanity over many hundreds of years, becomes more challenging or even impossible. So I'll close there and thank the people in my lab who have really spearheaded all of this work. They've worked very hard on many of these projects. I'll thank the funders, both Penn State and external funders, as well as my amazing collaborators here at Penn State who have made all this work possible. And I'll thank you and wait for questions at the end after Dr. Cotruvo gives his talk. So thanks very much.
Yeah, thank you, Charlie, for a great lecture. We are looking forward to questions, so I'm sure you have many. Please use your note cards to write those down, and we will collect them after Joy's talk. While we transition, I would like to share a few statistics. So Penn State was recently ranked as the #3 university in the US pursuing the UN Sustainable Development Goals by the 2025 Times Higher Education Impact Rankings. And in addition, Penn State was also ranked #2 in the US and #1 in the Big Ten for sustainability by the QS World University Sustainability Rankings in 2025. And I would like to think that work of Charlie and Joy have a lot to do with some of these rankings. Joy, you ready?
Okay, great. All right. So the problem is not so much having the rare earths in the US. We do mine them. The problem is the processing. And the reason why we, for a long time, did not process rare earths in the US is because we felt that scenes like this were not acceptable for environmental and public health reasons. So why do we have to, or why do scenes like this exist? It comes down to chemistry. And so, of course, as everything does, right? So let me introduce you a little bit to the chemistry of rare earth elements. These elements, sorry, I guess I should just stay over here. The rare earths are a series of 17 elements, yttrium and scandium, and then the 15 members of the lanthanide series down here on the periodic table. This is a periodic table showing critical elements. You may have heard the term critical minerals in the news as well. These are metals that are essential for particular really important technologies, and the rare earths are certainly a great example of critical minerals. So I think it's instructive to think about the history of these elements, because it turns out the names of these elements tell us really important things about the chemistry that, frankly, are still trying to be resolved. So 4 of the rare earths, yttrium, erbium, terbium, and ytterbium, sound like, very similar to one another. And that's because they were all isolated, in addition to other rare earths, from the same mine in a town called Ytterby. And this town is close to Stockholm in Sweden, which is why holmium is called holmium. And as I mentioned, all of these are in Sweden, and scandium and thulium come from the Latin and the Greek names for Sweden or Scandinavia in general. Similarly, so that tells us that all of these elements are frequently found together. Dysprosium comes from a Greek word for, difficult to obtain, and the term rare in the title rare earth elements reflects the difficulty of purification of these elements. Lanthanum, for which the whole lanthanide series is named, comes from Greek word for lying hidden, because this element was initially found as an impurity in a previously discovered rare earth called cerium. There was an element called didymium, which comes from the Greek for twin, and it was named because it was found along with lanthanum and cerium. But then it turned out in one of, I think, a very funny historical accident that the twin was actually 2 elements in the first place, the green twin and the new twin, praseodymium and neodymium. Okay, so these elements co-occur with one another in ores, and it took a lot of work 200 years ago, and still takes a lot of work to separate one from all the others. And this comes down to chemistry again, and because these elements, predominate in the +3 oxidation state, so they have a +3 charge on each of them, and their sizes are extremely similar. So this is a representation of the lanthanide series up here to scale of each of these ions. And so maybe you can tell the difference in size between lanthanum at the beginning and lutetium at the end But if you try to look at and find the difference between lanthanum and cerium, you may be hard-pressed to tell them apart. And molecules are hard-pressed to tell them apart as well. So when it comes time to mining these elements and turning them into technologies, we need to separate one from all the rest to extremely high purities, maybe 99.99% or so. So very high purity. And the way that that's done conventionally is in huge plants with many, many different stages of what's called solvent extraction. And solvent extraction involves an organic phase, often kerosene, and an aqueous phase, which is the rare earths and other metals as well in a very strong acid. So the processing of these elements uses a lot of nasty stuff, and it takes many, many, many stages, dozens or hundreds of stages. So this is really inefficient. It does work, but it's very inefficient. It uses a lot of just environmentally bad chemicals, and it costs a lot of money to operate these plants. And so I and many other people in this field feel that if the US is going to onshore its production of rare earths, including rare earth magnets, we have to take a fundamentally different approach. And so there are many people who are working in this space. The Department of Energy funds an institute called the Critical Materials Innovation Hub, which works on a number of critical materials, critical minerals, including the rare earths. I'm somewhere here, somewhere up there, I don't know. I know what I look like, but it's too hard to find exactly where it is. And Penn State also has a Center for Critical Minerals, and there are numerous people across in the Eberly College of Science and also in the College of Engineering, mainly, who are working in this space. But the reason why I'm here telling you about this work is that our approach comes from biology. And this is because it was found only 15 years ago or so that certain bacteria actually use rare earth elements. And these bacteria are really fascinating bacteria. They're called methylotrophs. And they're able to use compounds like methane and methanol as their only carbon source. So you and I need protein and glucose and fats. For our carbon sources, these bacteria only need, they can get everything from methane, some of them, and all of them can get it from methanol. And an important enzyme that they use in doing that is called methanol dehydrogenase here. So these bacteria are all over the place in the environment, but many of them grow on plants. And here's a plant leaf that's stuck on an agar plate that has methanol in it. And you can see that after sticking that plant leaf there, you see a bunch of pink colonies here of different bacteria that were living on that plant leaf. So plants produce a lot of methanol. It comes from the remodeling of the cell wall. So a link to Charlie's talk, specifically from pectin demethylation, I believe. And so as I mentioned, the enzyme that does this is called methanol dehydrogenase. And the big find in this field is that some of these methanol dehydrogenases require a rare earth to be inserted into them, such as the one here. Okay, and these bacteria can only use certain rare earths. Usually it's some mixture of these lighter ones, lanthanum through neodymium. So essentially, they're able to tell these apart in ways that are difficult for humans to do. And so at the time when my lab started 10 years ago, nothing really was known beyond the existence of these enzymes and that they use lanthanides or rare earths. And what we were interested in doing was seeing if we could discover some of the molecules that these bacteria are making that are involved in taking up the rare earths from the environment. And in so doing, perhaps we could come up with some more efficient ways of harvesting and separating these elements. And so we had some ideas, but there weren't really good starting points. And so we were growing some of these bacteria, these methylotrophic bacteria, in a medium that contained lanthanides. And what we isolated a protein that was in these bacteria under these conditions. And what we noticed from the amino acid sequence of this protein is that it had a lot of similarities to very well studied calcium binding proteins, such as calmodulin here. And what is shared by these proteins is a motif called an EF hand motif. It looks like a hand, and there are two helices. There's a helix going into the hand, which is where the metal binds, the calcium binds, and then there's another helix going out of it. So you and I have dozens of proteins that look like this, dozens of different proteins that look like this. So we had a suspicion that this protein was going to bind some rare earths, but the thing that cinched it is we did an experiment where we looked at the amount of these helices that are formed when we add in different metal ions to the protein. And you can see that all of the rare earths form this helix, but then, interestingly, when we added calcium to the protein, nothing very much happened. And the reason that's the case is because it turned out that the structure of this protein was totally different from other EF hand proteins. And so this is a representation of that. The spheres are metal ions here, and in other structures that I'm going to show. And you can see the helices going in and going out of the metal binding sites. So it turned out that this protein was extremely good at differentiating rare earths from everything else on the periodic table. So actually, it was so good that we had to turn to radioactivity to show how good it was. And so this particular experiment, which was done by a close collaborator at Lawrence Livermore National Lab, used actinium, which is one of the rarest elements on the periodic table and is a surrogate for a particular rare earth. And what our collaborator found is that the protein can withstand about a trillion-fold excess of other elements that typically will bind to other proteins. So this was pretty incredible to us. And schematically, what we believe is happening is that the protein is very floppy when nothing is bound to it. But when rare earths or lanthanides bind to the protein, it folds up into this state and it holds on to those metals really tightly. Other metals will bind to the protein in the floppy state, but they don't induce this tightly bound conformation. And this is where the selectivity comes from. So we had this hugely selective protein, and we wondered what we could do with it. And this applied work, much of it was done in collaboration with researchers at Lawrence Livermore National Lab. So initially, we found that, well, if we're going to investigate this protein as a technology, potentially, we need to make sure that it is extremely stable under harsh conditions that are necessary for separation processes. And it turned out that the protein is very stable. You can put it in very acidic solutions. It will hang on to the metal ions to a very low pH. You can also heat up the protein to 85 degrees Celsius or more, and it will hang on to the metal ions. These are pretty incredible properties for a protein. And so we imagined that we could have the protein exposed to a number of different metal ions, including the rare earths, and then physically separate them and see what was bound to the protein and what was unbound from the protein. And we started by looking at a leachate of electronic waste, such as old cell phones, that has a lot of rare earths in it. And when we did this, what we found is that the protein depleted the rare earths from this electronic waste generated solution, and basically depleted all of the rare earths, 99.5% of the rare earths, and it seemed to bind almost nothing else that was present under those conditions. So then again, thinking about how we could make this technology perhaps more scalable, if we can immobilize it on a surface and put it into a column, then we could run through solutions, see what binds to that protein that's immobilized, and then have everything that doesn't bind flow out. And so we figured probably we would be able to get the rare earths to bind to the protein and everything else to flow through. And so to investigate this, we started with coal fly ash, which turns out also has a decent amount of rare earths depending on its source. So you can leach this and pass it through the column. And what was observed is that everything that's not a rare earth pretty much flows through the column. And then everything that's a rare earth up to a certain point, when the column gets saturated, binds. And it turns out we start with about 0.04% of the material in this solution was a rare earth. And just by passing it through the column once, we increased the purity of that solution to almost 90%. So about a 2,000-fold upgrade in purity by doing this. So then it turned out that initial protein was not so good at separating the rare earths from one another, which is really the hardest problem. So we went back to biology and said, well, we think we know how to identify what is this landmodulin. I never told you the name of the protein, although it was on the slide. I forgot to do that. So landmodulin is what we called this protein. We felt that we had a good idea of what would differentiate landmodulins from every other type of protein. And this is a representation of basically the similarity between all of these different proteins that we thought are landmodulin proteins. So this is the one that I was telling you about before. And then we looked at a protein that's over in this space over here, far away from this initial protein that we looked at. And we called this one Hans because, as you can tell, the name of this organism is probably a little bit hard to say. So Hans, also it's nice to think of these proteins as people in a sense. They have identities, they have personalities. I don't know, maybe that's a little strange to maybe you don't like thinking about proteins like that, but I do. So Hans was really interesting because not only did it fold up into this tightly bound structure when rare earths bound, but when certain ones bound, the protein also interacted with another one of itself to form this dimer. So the lighter rare earths, lanthanum, neodymium, etc., induce this dimer, but the heavier ones, like dysprosium, do not. The protein stuck as a monomer. So why is that? We got the crystal structure of the protein. That's what's represented here. One of the metal binding sites is right at the interface between these two monomers. And there's a lot of interactions between those two proteins here. And if we zoom in, we can see what differentiates the protein when it's bound to a light rare earth like lanthanum and a heavy rare earth like neodymium. So with a light rare earth, which is a bit bigger, the protein is reaching out, specifically this residue is reaching out with both hands to grasp the metal ion, the lanthanum ion. But dysprosium's a little bit smaller, and so both hands can no longer grab. So now when it's trying to reach out, it can reach a little bit further if it twists a little bit and one of the hands grabs the ion. And so that's what we're seeing is happening here. And the result of that small shift is that it initiates a cascade that makes the interface fall apart, and the proteins no longer interact with one another. So I think this is a nice example of how you can take really small differences in the size of these ions and turn them into really big differences in the molecular properties here of these two proteins. So in this case, dimerization. So then we were excited about this differentiation mechanism and if we could use this for separations of rare earths. And so here we're doing the same thing. And the idea with immobilizing the protein and putting it through the column is first we will get a purified, or we hope to get a solution of purified heavy rare earths because they bind a little bit weaker to the protein. And then we can elute with a dilute acid and purify the lighter rare earths that bind a little bit tighter. And so initially we started with neodymium and dysprosium. These are rare earths that if we're going to recycle electronic waste, we need to be able to do this separation. And now this protein does a really great job of separating the dysprosium, which comes out first here, and the neodymium that comes out second. Furthermore, if we consider, a real world feedstock is going to have many more rare earths, especially if we start at the mining stage rather than the recycling of waste stage. So being able to differentiate much closer rare earths is really important. And even in this case of gadolinium and dysprosium, so here's dysprosium and here's gadolinium, we get pretty good separation of these two metal ions. So it turns out we can apply an even more complicated solution and get rare earths to 95% or greater purities from these experiments. So what we envision here is replacing the solvent extraction step with a purification step, separation step that uses this protein in some way, perhaps including a column. And I can mention that we had a patent application that was recently issued on this technology. There are two companies that have licensed it and are scaling it up and developing it further, and we're excited to see what happens. So today, hopefully, I showed you that biology makes some really amazing molecules. They have really, really high selectivity for rare earths. Lammodulin is one of them, but we've discovered some other proteins as well that have interesting behaviors that we're using for separations as well. And what we envision here is that we can merge the selectivity that comes from a biological system with the scalability that comes from chemical approaches, such as the one that I showed you with the column immobilized protein. And this also raises the question of whether analogous approaches can be used to harvest and separate other critical minerals. So I'm grateful to all the people in my lab and our terrific collaborators, including collaborators at Penn State, who have contributed to this work. There's a disclaimer for you to read, but you can believe all the things that I said. Thank you for your attention and look forward to the questions.
Thank you to Charlie and Joey for those awesome lectures. While Wansuk is collecting more cards, I do have a question for both of you. So a lot of my friends, outside of science and academia, whenever they hear about our work, they always think about the ecological impacts of it. So my work, Charlie highlighted it a little bit, is with wood. And so the question that they ask me all the time is, okay, if we chop a bunch of trees for ethanol, wouldn't that have ecological consequences? If we mine a bunch of, rare earth elements, wouldn't that also have some sort of, downside on our ecology? What would you guys answer to that question? How would you appease my friends?
Thanks, Elisa, for that question. And one of the things that I always highlight is the scale, the relative scale. So we have to think about how much of a given resource we're harvesting at a given time. There have been a series of publications from the Department of Energy called the billion-ton vision, right, which is basically using 1 billion tons of biomass to generate about 1/3 of the total fuel use of the United States. I contrast that with how much biomass exists, right, in the United States, which is probably in the 100 billion tons range. And so if we're harvesting 1% of the lignocellulosic biomass to generate fuels, that's actually probably lower now that electric vehicles are taking off because of rare earth elements and other things, right? We still probably are going to use fuels for the short term, at least for things like sustainable aviation fuel, shipping fuel, things like that. So we want to do any kind of biotechnology and use of a resource in a way that is sustainable and allows that ecology to regenerate it without being depleted, right? That's true of basically all of resource utilization across the economy. So that would be my sort of answer to that question. And we have to be thoughtful about it and not say we're going to just harvest all of the wood in the world to make biofuels. Unfortunately, that has happened in some places like Malaysia, where there was a big push in places like Europe to implement biodiesel as a fuel, and that incentivized farmers in Malaysia to basically convert large parts of rainforest into palm plantations, because palm oil is one of the main sources of biodiesel. That was devastating for that ecology and led to drops in the population of things like orangutans, which are very charismatic organisms. And so we do have to be thoughtful about that and do it in a way that incorporates into our existing economy. So convincing farmers that maybe growing switchgrass on a portion of their fields that might not be especially productive for food would be a thing to do, and not doing it in a way that really depletes the ecosystem.
Yeah, so from the mining perspective, I mean, I'm a biochemist, so by training, so I've been learning about mining along the way. But I guess I will say that the U.S. used to be the world's main producer of rare earths until the 90s, I believe. And one of the reasons why we stopped doing that for a while is the environmental issues that came out of that by doing mining from conventional approaches. And so the strategy is, well, we'll just offshore that because we don't like the environmental downsides from that approach. And that maybe works for a little while, but it doesn't really solve the problem in a long-term approach. So long-term, we need to change the way that we do things. And so right now, there's a lot of investigation into, if we mine rock, how can we use all of the elements that are present, or as many as possible in that rock that we mine? Or how can we very selectively extract one element or one type of element from a particular source, and then put as much rock back into the ground as possible to minimize the environmental challenges that are associated with that? With rare earths, it's challenging because rare earths are often found with a lot of uranium and thorium, so radioactive elements. And that leads to a lot of management issues, as you can imagine. So in that case, putting as much back into the ground is maybe the best approach if we can do that. So I think people appreciate now much more than maybe they did 30 years ago that if we are really going to use these sources, there's no way around around mining. Like, we have to mine if we want the elements for these various technologies. Recycling is great, but there are a lot of challenges with recycling, and recycling is not going to meet the demand that is only growing for these elements. So how can we do this in a responsible way? A lot of people are thinking about that these days, including the mining companies. Yeah, that's what I'll say.
Thank you. Okay. Two questions for Charlie. Could you differentiate between plant and animal cells? And second question, do digestible plants have different cell walls?
Yeah, that's a great question. So animal cells and plant cells have a lot of common components. They both have DNA and a nucleus. They both have microtubules. They both have mitochondria. Plant cells are unique in the sense that they have what are called plastids, like things like chloroplasts, which allow them to photosynthesize. We think that this happened about 1 billion years ago when the ancestor of a plant cell engulfed a photosynthetic bacterium that was called through a process called endosymbiosis, and gained the sort of superpower of photosynthesis. And one of the other big differences is that plants produce these polysaccharide-based cell walls. So many of our cells produce what's called an extracellular matrix that's largely composed of glycoproteins, but it doesn't contain the same polysaccharides like cellulose and hemicelluloses and pectins that plants are able to generate. What that ends up causing is the plant cells to be sort of locked together into a brick-like framework, whereas in animals, the cells can move around, which is beneficial to us for things like our immune system, our cells being able to navigate through our body. Plants have a different type of immune system, partly because their cells are locked to each other, which was about what differentiates digestible versus indigestible parts of plants. Part of it is the content of cellulose and lignin. More cellulose and more lignin makes plant cell walls less digestible. So think about, the difference between eating a fruit, which is largely composed of pectins, versus a stem, a branch, right, which is largely composed of cellulose and lignin. So that's one of the major differences. Our bodies are pretty good at digesting hemicelluloses and pectins, not so much cellulose and lignin.
And then for Joey, when rare earth metals are used up, do they lose their magnetic property?
So demagnetization is part of the process of disassembling the rare earth magnets in electronic waste. And so yes, unfortunately, what has to be done is you go all the way to the component ions, and then you purify them, and you make metals again, and make the magnet again. So recycling of magnets is challenging. People would love it if you could just start with the electronic waste, get out the magnets as is, and put them back into new technologies. But unfortunately, in part because of the challenges of just how we collect electronic waste, that these magnets are part of, the electronic waste itself is a very complex mixture of things, even in a single device. There's no way that people have found, at least so far, of skipping parts of that process.
Great, thank you. This questions are sort of related to mine, the ones that I asked. Are the byproducts of bacterial rare earth separation processes toxic? If so, how do you safely dispose of them? And if not, are they useful for something else?
Yeah, no, just taking a moment. So the byproducts of making, so for our approach, which is producing protein, using bacteria to produce proteins, immobilizing the proteins, and then using them in a separation process, the protein production part, there's this is pretty standard for production. Proteins are produced industrially for high fructose corn syrup production, for example, all of our favorite things. We love high fructose corn syrup, right? But yeah, so there are wastes, but I don't believe they're any really toxic in any way. So from the process itself, utilizing the protein for separations, acids are involved, at least in the process that we're using at the moment. Obviously, we're trying to utilize as little acid as possible, and we don't have to use any organic solvents. The kerosene part of the solvent extraction step. The idea is that we should be generating much less toxic waste from our process versus the standard. Now, this takes a lot of analysis, a lot of economic analysis and life cycle analysis to be able to really implement this. And this is work. This is one of the things that the companies are doing in thinking about how this technology can be scaled. But that's a great question.
Thank you. All right, this next question is for Charlie. Are superwoods any more resistant to fires?
That is a great question that I don't think we really know a convincing or complete answer to at the moment. I will say that we've collaborated with David Hughes, who's a researcher working in sort of sustainable food production here at Penn State, along with partners in Africa, to send some of our superwood to Africa to see if it's termite resistant. So that's probably the closest answer I have at the moment. The density of that wood, I would hypothesize, would make it more fire resistant just because it's more dense. But it probably will eventually burn. It is also possible that you could generate superwood as a highly dense fuel that might burn cleaner than non-densified wood, but that's, I think, still to be determined experimentally.
Right. This next question is for Joey. What is the potential of designing novel enzymes with AI that could purify rare earth metals?
That's a great question. So I guess one technicality is that we're using proteins, and some proteins are enzymes, but not all proteins are enzymes. And enzymes catalyze chemical reactions. We are just using proteins to bind and release metals. So anyway, AI. This is one of the things that we are investigating. Actually, my sabbatical a couple years ago was in David Baker's lab, who won the Nobel Prize a couple years ago, I guess, for protein design work. And yes, so for lanthanides specifically, or for rare earth specifically, it's an interesting question because we just don't have that much information that can really be used to train the AI algorithms to get good to be good at redesigning selectivity, for example. But I think this is a really important way forward here to get away from, the natural systems have evolved in a particular way. And some of their selectivity is amazing, way better than what's possible or what's known with other systems. But the pressures of their evolution were not the same as those that we are under, right, as humans, and the reasons that they have made these proteins are different from the ones that we want to use. So I think AI will be very valuable, but you always have to be smart about it, right? I think we want to make sure that we don't go too AI crazy and stop thinking about about anything, right? So yeah, that's just an important thing to keep in mind. But yeah, it could be very exciting.
So I have an add-on comment for that question, which is that we've also explored the use of AI for designing new cellulose degrading enzymes in collaboration with Costas Moranis, who works on the 3rd floor of this building, I believe. And so far, the AI designs for cellulose degrading enzymes have been kind of slop, I have to say. Enzymes that degrade cellulose in fungi and other organisms have evolved over, again, hundreds of millions of years to allow those fungi to survive on, let's say, a log, right, in the forest. And they are not optimized necessarily for degrading the cellulose as rapidly as possible because in that case, the fungus is degrading its own food source. And so we think there are opportunities, but we have to, again, be smart about it and really complement our own creativity, our own human creativity with the tools that AI provides for protein design.
Great. Thank you. This next question is for Charlie. Has superwood been used to make musical instruments, for example, guitars? How do they sound?
That's a great question. I've had like daydreams about building a guitar. Elisa, no pressure, because it's no pun intended. So superwood is makeable, but you would need very specialized equipment to do that. So maybe that will be something that Elisa can make, for her PhD dissertation and learn how to play a guitar at the same time. So we have not tried that yet. However, on a slightly more serious note, one of the major companies that produces guitars is based in Pennsylvania. It's called Martin Guitars. So if you've ever heard of a Martin guitar, they're very famous and very expensive. I do not own a Martin guitar because I can't afford it. But I've thought about approaching that company to say, like, could we actually take something that's a Pennsylvania native hardwood and make it into something that's more like a tropical hardwood by doing this kind of densification process? The challenge, I think, would be maintaining the structural integrity of that material and also the sort of timber that's based in part, Timbre, not Timber, that's based in part on the air spaces within that wood porosity, right? So those are things that we'd have to think hard about. Maybe we can make some sort of micro guitars to start out with and see what we get. Yeah, ukulele, that would be a good starting place. Thanks, Sasha.
All right, this one's for Joey. Where have the bacteria that land modulans have been isolated in, were they found in any extreme environments?
Yeah, so there are, so the bacteria where land modulans are predicted to be from, including the ones that we've studied, are not from extreme environments. The Methylobacterium extorquens, which is where the protein was initially found from our studies, that is one of the bacteria that grows on plant leaves and is associated with root systems, I believe, of some plants as well. Now, there are other bacteria that have been found in more extreme environments that are able to use lanthanides. And in fact, the first bacterium that requires lanthanides for growth was isolated from a volcanic mud pot, in a really cool story where they went to the mud pot and got some water and then tried to culture that bacterium in the lab, and they couldn't do that until they went back to the source and eventually figured out what it was in the mud pot water that was allowing the bacteria to grow, and it was the rare earths. So the solubility of rare earths is pretty high in the acidic environments and hot environments of a volcanic mud pot. So it's possible that that's where this evolved in the first place, this ability to use the rare earths.
Great. Thank you. I think this question might be for both of you. Are there any plants that can draw out poisonous chemicals out of the dirt as they grow in this dirt? And if so, how?
You want to, more about
maybe.
plants
Yeah,
than I
you're
do,
welcome to
so.
follow up. Yeah, so this is an area of research called phytoremediation, right? The idea that we can use plants to draw toxic chemicals, toxic metals, even things like polyfluorinated alkyl substances or PFAS, which are called forever chemicals, out of the soil. I'm collaborating with somebody in engineering named Honor Apul, as well as a researcher at Kansas State, to actually test out some of those ideas using various plants. So this is an active area of research. I think the jury is still out whether plants are going to be the answer for remediating soils that are contaminated either with metals from mining or things like some of these synthetic chemicals that we've made that are causing harm in the environment and harm to humans. But that is certainly one strategy. And plants, in some cases, are pretty good at what's called bioaccumulation. They're able to draw these things out of the soil and cause them to be concentrated in specific parts of the plant that we could then take and process or even do something as simple as burning. That's one of the sort of methods for getting rid of PFAS, for instance, is just incinerating it. So if we were able to do that, that might allow us to remediate some of the contaminated soils in Pennsylvania and elsewhere. I don't know if you have anything to add. Okay.
Pretty complete.
Thank you. Another question for both. Do either of you think that industrial espionage would be a big concern?
That is a good question. So at Penn State, you might have heard the recent town hall from Dr. Andrew Reed, who's our senior vice president for research, in which he highlighted the concern about what's called export control, meaning some of the ideas that we are generating here at Penn State, making sure that those are protected in ways that are beneficial both to Penn State, to the inventors, and also to the United States, the interests of the United States. And that is something that we have to be a bit, I would say, a bit more cautious about in today's geopolitical climate. When we travel to other countries, I recently traveled to China, for instance, and I had to undergo a review to make sure that all of the science I was talking about was what I would call public science, right? Science that was not going to be patented, but that was just going to be published as kind of fundamental science. So you might have more thoughts about this since you've actually started companies and had IP generation in your lab.
It is an issue, definitely. The government is very aware of these issues, particularly in the rare earths space. Part of our research is funded by DARPA, and they have additional levels of they review everything that we put out in presentations and in papers. So yeah, this is definitely on the minds of the government. I get reached out to occasionally, I have to say, and I do not respond to those to those requests when they come from certain foreign countries. So yeah, that's the reality of the time. I'm happy to say more off the record.
All right. This one's for Charlie. Could you comment on if there are any downsides to producing corn ethanol and how that has an impact on potentially soil erosion?
Yeah, that's a great question. There are downsides to any technology, right? Any technology has upsides and downsides. And so one of the challenges with producing corn-based ethanol, which has been very successful as a biofuel in the United States, for instance, because we produce a lot of corn, is that it, as I mentioned during my talk, it generates this competition between corn as a feedstock for ethanol production and corn as a feed, right, for animals, or corn as a food for humans. And so that can generate some market imbalances that lead to increases in the cost of food that are not beneficial for both Americans and especially people living in countries where growing corn is a subsistence activity, right? Because then the incentives become more to sell the corn than to consume the corn themselves, and there are problems that can arise. Corn, like many crops that we currently grow in the US, requires a lot of inputs, meaning that we have to provide a lot of fertilizer and, in some cases, irrigation to grow that corn. Not so much in Pennsylvania and in the Midwest, which have abundant rainfall. But one of the challenges that arises with growing corn and soy, right, sort of rotating corn and soy, which is kind of the typical agricultural practice here in the US, is that by adding all of those fertilizers to the soil, when there are extreme rain events or even just regular rain events, we get leaching of those nutrients out of the soil and into our waterways. So you've probably heard of this sort of phenomenon of so-called eutrophication, where these fertilizers get into our waterways, they enter places like the Gulf of Mexico, and that can lead to harmful algal blooms that deplete oxygen from the water and really disrupt the ecosystem of places like that Gulf. So that's one of the challenges. Soil erosion is just a constant challenge with agriculture wherever you are and whatever you're doing, because soil is a resource that, if you do agriculture really carefully, can be replenished every year. But if you do agriculture in a way that's extractive, does lead to erosion. We've had that happen in phenomena like the Dust Bowl back in the 1930s. So people are aware of these challenges. It's just a way, I think it's a matter of implementing smart policies that don't incentivize farmers and other workers in the agriculture and forestry sectors to do that kind of extractive and destructive work on the land that they really do care about a lot.
All right. And for Joey, is it possible to source rare earths from acid mine drainage in Pennsylvania coal mines?
Yes, and that's a great question. There are a number of projects that have been funded to look into that, including some at Penn State. With acid mine drainage, well, so you probably know that whoever asked that question probably knows that acid mine drainage does have rare earths, and interestingly, the concentration of the heavy rare earths, which are the ones that are more valuable on a like per kilogram or per ton perspective, those are relatively enriched in acid mine drainage as a part of the mechanism of the leaching process. And so there is interest in doing this. The challenge is that most sources, the concentrations of the rare earths are very low. And the volumes at which the acid mine drainage is being produced are very high. So there are very few technologies that can really work under those conditions. And because that's the first challenge. And the second challenge is that because the concentration is so low, you need to go through so much material to be able to get a saleable amount of the metals. And it's just challenging economically. So there are methods that work to do that. The proteins, for example, some of our applications have involved sensing rare earths in acid mine drainage. But yeah, the economics, I believe, are just not there at the moment, even in the best cases.
Thank you. This question's for Charlie. Does Penn State collaborate with the U.S. Forest Service Research Lab in Madison, Wisconsin? It's related to the wood work.
That's a really good specific question, not to my knowledge at the moment, but we are reaching out to potential collaborators to work more on these types of projects. There's a really amazing forest in Wisconsin called the Menominee Forest. It's owned by the Menominee tribe and is used to do sustainable forestry, meaning that they harvest from that forest in a way that is economically sustainable and also sustainable for the ecology. So my hope is that foresters here in Pennsylvania and around the country will learn from people like the Menominee to do forestry in a way that is both profitable and sustainable.
Great, thank you. Another question about corn for Charlie. Do you know anything about the use of native corn varieties? So for example, from the Southwest in ethanol and food production?
I don't know a lot about that because I'm not a corn breeder myself, but I will say that most of the corn that's grown in the US is not a native variety. It's a more engineered variety. Many of the corn much of the corn that's grown currently is what's called hybrid corn. And that is beneficial for a yield, right? So hybridization of corn does provide a benefit for yields, with the downside that then or sort of trade-off that corn is then has to be sold every year from a seed company to the farmer. So it makes it harder to do seed saving, which is a practice that is less common now among farmers. I think there is a recognition that many varieties of corn have beneficial traits, and there are organizations that are working to preserve and save and expand existing varieties of corn, including some of these native varieties, in ways that will benefit agriculture into the future.
Great, thank you. Just one final question for the both of you. So both of your work has some sort of, application, right? So this question is asking, what are the policy implications of your work? And in other words, why should anyone care about what you're doing? Is scalability important to your work?
Well, I would say if you like to wear clothes and eat food and live in a house, you should support my research. But sort of again, slightly more seriously, I think the bioeconomy is going to be a really important part of the United States economy going forward. We are incredibly blessed to have both amazing mineral resources and amazing bioresources here in our country. And if we do that in a smart way, I think we can maintain a position of economic competitiveness with our neighboring countries around the world, in a way that's beneficial both for us and for the global economy. So I would say that, understanding and building the next phases of the bioeconomy is going to be one of the really important research efforts of this century.
So in terms of mining and bioeconomy, traditionally, biology hasn't had too much to do, and biochemistry hasn't had too much to do with the mining industry. Copper mining being a very important exception there. There's, I don't know the number, but something like 10 or 20% of global copper production does come from a process that involves organisms. But this is something that the US government is very interested in these days. I was just a couple months ago, I was co-chair of a Department of Energy workshop that was specifically focused on how we can use biology and bio-inspired ideas and bio-inspired ligands or molecules, I should say, for critical mineral recovery. So not just rare earths, but extending to many other important resources. And so I think as the appreciation for what biology can do and how we can interface it with chemistry and material science and other sciences to solve some of these problems in novel ways and perhaps more sustainable ways is something that's getting a lot of attention these days. And I would like to think that my lab's research has helped give the government a little bit of a kick in the butt to move in this direction. But of course, it's the work of many people that's gone into raising the profile of this type of work. And so we'll see. There are a lot of challenges, right? Scalability for any new technology. Scaling is critical. But fortunately, there are a lot of other applications of biotechnology that you can draw from in doing the scaling of these sorts of mineral focused technologies. So we'll see. I think it'll be really exciting in the next decade, 2 decades to see what happens.
Great, thank you so much. So we are out of time for the Q&A session. I want to thank Charlie and Joey for answering all of your wonderful questions.